Okay, so what we have here in general, I'm going to have to draw a kidney picture here, right? In general, I won't flip back to that picture we had of it there. You have not only this single tube that we called the ureter coming out of a kidney. That tube is draining all the nephrons, right? You have one big blood vessel entering the kidney. That's bringing the blood to all those glomeruli, all those capillaries and glomeruli. So this one blood vessel is going to branch into literally a million different branches, a million different glomeruli. This would be called the renal artery. It's taking blood away from the heart and toward this organ. In another chapter, we'll focus on the fact that this blood also has oxygen. And this is delivering oxygen to all these cells, but now we're focusing on a different function of the kidney, right? So this blood gets filtered out, and the blood that now has had a good bit of urea taken out of it is now going to have to leave the kidney by a blood vessel that's going to take it on its way back to the heart. And veins are blood vessels that are on their way back to the heart. Renal's the kidney word. And so they might ask you an open-ended question. How does the composition, and by that they mean what kind of things do you find in it? Water, ions, stuff like that, urea. How does the composition of blood that's coming in the kidney differ from blood that's leaving the kidney? And your simple answer is it's probably the blood coming in the kidney probably has more water in it because the kidney's going to kind of filter some out. Sorry, I should take that. Sorry, let me slow down, right? Because what they could also add into this story is how does the composition of the blood in both of these arteries compare to the composition of the fluid in the ureter. There's really three tubes that here have different composition, and they could easily factor that one in, but they threaten you specifically with comparing the renal artery and renal vein stuff. So what did we, this kind of then gets simple. What did we take out of the blood that's coming in? Urea. So the urea content in the renal artery you would predict would be higher than the urea content of the renal vein. Where did it go? It went into this, right? That's going to end up in the ureter. So then when they ask you about sugars and salts and things, I think you would predict probably pretty much the same, because those useful things like sugar and salt and water itself that you might have taken out of the blood in filtration got put back into the blood. And so I think you'd predict everything but urea to be about the same in the renal artery and vein. But compared to this, compared to the what we would still call the filtrate and now call urine, right? We can call those the same thing. A much higher urea content in the urine, in the filtrate, in this tube, right, the ureter, and much less of it in this blood vessel compared to both that blood vessel and the ureter. So here they also ask you like to predict, if you live in a very dry place, what might you predict about the ability to concentrate urine? You would predict that, well, they can probably do that a whole lot better. How might they do that a whole lot better? This is coming up in the next slide, I think. Well, what kind of part of this system allowed us to squeeze a lot of water out of that filtrate and put it back in our blood? The loop of Henle, right? So what would you predict about things that really do that well? A very long loop of Henle. And if you do some dissecting, your prediction would be verified anymore. But there it is, ADH. Okay, ADH is a hormone. That's what the H stands for. Antidiuretic is the AD part, right? This hormone is produced in a part of your brain that has cells that can sense how watery the blood is. And when they get the message that your blood is a little too hypertonic, just to throw those words in, not watery enough, they secrete this chemical, right? Actually, sorry, I'll add this because we're coming to this too. I oversimplified there. Your hypothalamus is a part of your brain that has the cells that sense that. They send a little different signal molecule to cells in your pituitary gland, and they are the ones that are going to send this ADH out into your bloodstream. And in your bloodstream, eventually they're going to bump into cells that have the right receptor that this fits. So this is also part of the story. We have signaling from a long distance here, signal molecules from a long distance. That's in the AP syllabus, right? And when they hit these cells in the collecting duct, they cause them to be more permeable to water. So that helps water get out of the collecting duct, which is on its way out, and back into your body. And again, we emphasize that this is the last chance where this filtrate goes through a tube whose walls are thin enough for stuff to get in and out. Okay, so that makes your fluids in your body more watery, right? But whether they're watery enough to tell your brain to turn this negative feedback system off really also depends on, you better drink some water, right? Because squeezing extra water out of your urine can only help you keep water to a certain degree. You better keep drinking water if you want to actually hydrate yourself again. Okay, so some other dynamics of this. How this works is maybe on the next thing, but I'm gonna use this picture because I got a lot of space to draw. If this represents a cell that's in the lining of your collecting duct, this would be like transport epithelium. In fact, I should have said that word. All those tubes, the capillaries and the glomeruli, all the nephron tubes, they would all be called transport epithelium. The tissues made of cells, right? So what these cells have inside of them, it turns out, is vesicles that have aquaporins in their membrane, right? And when on these cells, I'll make a simple receptor, this ADH is the right shape to fit into this receptor, signal transduction pathway, causes cytoskeleton in this cell, we're reviewing a lot of stuff here, to move these vesicles to the plasma membrane and fuse with it. So now these aquaporins that are part of the vesicle membrane are part of the outer cell membrane, and that's how this cell can have water in the tube. I'll put some of them here. Go through aquaporins and get out of the tube. This turns out to be the exact same mechanism we'll talk about in a chapter later. The exact same mechanism that glucose transport molecules get moved in a cell from being part of vesicles to actually being in the plasma membrane to let glucose in. And the hormone that attaches to a receptor and triggers that glucose transporters being actually added to the plasma membrane is that familiar hormone, insulin. Okay, so anyway, all that can be part of this story. Why is this negative feedback? Why is this negative feedback? And here you see in the IB thing, they want you to specifically know ADH and what it does, right? They also expect you to know where it's secreted from, the hypothalamus or the pituitary. Either one of those could be a correct answer to that. They're actually, when we get to nervous system, we'll see this. They're actually physically connected. They almost shouldn't have two different names, but anyway, they do. So why is this negative feedback? Because if the message is, or the situation is, you're low in water, that low in water itself, that dynamic itself, stimulates a mechanism to happen that causes you to get higher in water. And that's, again, always the idea of negative feedback. whatever the direction of the stimulus, in this case lower water, the response is to turn that in the other direction. Okay, so this is what I said, right? This is what I said about insulin and glucose channels and da 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 da, right? So this is all what I just said. So you can stop it and read if you want. Okay, so I'm going to throw this in. This is not in either syllabus, but just because, and we probably talked about this already in class, that a protein called ACE2, it's actually an enzyme involved in blood pressure. And part of that is also, blood pressure is also how much water is in your blood, right? Because your blood is mostly water, and if there's not enough water, then your blood pressure is a little too low, right? Well, if your blood pressure gets too low, then another kind of hormone is produced, which this enzyme converts to a different hormone, all of which you don't need to know the names of, right? These would be the these hormone names. And this molecule doing this and this causes also the same kind of increase in squeezing water out of your blood to help raise your pressure, right? Which is why if you have high blood pressure, some medicines you might take block this, attach to it so it doesn't work, because what this does is increase your blood pressure, right? And that's what initially made those kind of drugs ACE blockers, right? ACE blockers, hopeful treatment for the coronavirus, because we learned early on that the coronavirus attaches to this receptor, and that's how it gets into your cells and infects your cells. So anyway, with the immune system, we'll probably talk about that too. So here's that whole negative feedback loop involving ACE. I don't think they actually put this here. The ACE2 thing and all that. So I'm just gonna, you don't need to know this whatsoever. However, again, if you're told the story that I just told you, and it's kind of on the last page about in response to low blood pressure, this gets released and this gets released, we can connect lots of different questions to that, right? What are these molecules? They're proteins, right? So what would trigger a cell to make more of this protein at this point in time compared to another point in time? Some kind of signal molecule, right? Or in this case, maybe the drop in water triggers a signal transduction pathway in cells, just like a signal molecule would, that that is part of this mechanism that has caused the genes to make these proteins to be used more. So now, again, we're in the gene regulation category, which means we're in the transcription factor category. We're in all of that stuff. So, you know, again, as we get further and further in biology class, all these things we talk about get connected back to things we talked about before.